Okay, so in the mode of actions of enzymes, we have to always mention few terms that we have. I mean, two. Okay, we have no choice. First, we talk about substrates molecule. We talk about the active sites. We talk about the ES complex. We talk about the product. Means that we explain the entire process from the substrate, how we get the product. That is means of the mode of actions. Okay, are you clear? Okay. So how I explain now? First, I explain in general first. Later, I will show you guys in the more detailed way to explain. Depends on the question asked. Okay, so in enzymatic reaction, the substrate molecule is held by the active sites by a weak interaction. Okay? Bonds, in this case, highlights hydrogen bonds and ionic bonds. So these two types of bonds is very, very important. And how these hydrogen bonds and ionic bonds can be formed between the active sites as well as this between the active site and also the substrate, it totally depends on the R group that form the active sites. Okay, the amino acid, the R group, the amino acid that form these active sites. Okay, so if we look at these diagrams, then we have to be careful. We have to be careful. Now, why to be careful? Because in the exam we have to look at it, observe first my substrate. And second, my active site. What we observe? We observe whether the shape of substrate, are they exactly complementary to the shape of the active site or not. Be careful, guys. Be careful. First, you look at the shape of the substrate. You look at the shape of active site. Are they complementary to each other or not? If they're exactly complementary to each other, then we explain it simply. I mean, the way we explain it is more simple. So first, I'm going to say that. Okay, first, the shape of the substrate is exactly complementary to the shape of the active site. First point. Second point. So substrate bind to the active sites or the enzyme. Don't say bind to the enzyme. Bind to enzyme, it can be which parts of the enzyme. Are you clear? You do not know. So always remember, in A level, you must be precise. So first, I say that the shape of the substrate is complementary to the shape of active site. So therefore, substrates bind to the active site. Can you see that? The substrate bind to the active site by forming temporary bond. Do not need to mention hydrogen bonds or ionic bonds. It's okay. Just say that it forms temporary bond, general. So therefore, ES complex can be formed. Okay, so the formation of the ES complex, okay, in this case, lower down the activation energy. Because the temporary bond is formed, lower down the activation energy, reaction take place, and then products can be released, enzyme can be reused. Simple in this case. Because why? The substrate, the shape of the substrate is complementary to the shape of active sites. Okay, so I explain again. The shape of the substrate is complementary to the shape of the active sites. Therefore, substrate bind to the active sites, okay, by forming a temporary bond. Okay, activation energy is lower. So ES complex can be formed, reactions can take place. Therefore, in this case, hydrolysis can take place, then allowing the products to be formed, and products can be released from the active sites. Therefore, the enzyme can be reused. Okay, clear or not, guys, so far? Okay, so I pause the recording now. So first thing, we do know that the active sites of the sucrase is enzymes, okay? So active sites of the enzyme molecule are specific to its substrates, okay? My mistake here, it should be ITS, okay? It's substrates, which is sucrose, okay? And then the shape of the substrate molecules are complementary to the shape of the active sites of the enzyme. So highlight the word. You must include the word shape of the substrate complementary to the shape of the active sites. Okay, not other thing, it's the shape. Okay. So next, so substrate molecule bind to the active sites of the enzyme, forming enzyme substrate complex or ES complex. So they form the temporary bonds. So these temporary bonds can be hydrogen bonds, can be ionic bonds, are formed between the substrate and the active sites, therefore lowering the activation energy. So you do not need to mention a correct orientation, blah, 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 because that is a precise mechanism. Okay, unless question asks for the precise mechanism, how the enzyme lower down the activation energy, then you talk about that. Okay, then products are formed. In this case, the glucose and the fructose can be formed, and then released enzyme can be reused to catalyze the reaction. So this one, we use it onto the enzyme. They have exactly complementary shape. Are you clear? Okay, so always remember first step. Look at this. The shape of the substrate and the shape of the active site. Are they exactly complementary or not? Now, if let's say the shape is not exactly complementary, then that one is the induced fit model already. So when we explain the induced fit model, so this is how we explain, yeah? Look at this diagram. Look at these diagrams, yeah? So let's see. This is enzyme. Okay, this is a substrate. Okay, how I draw it. Okay, let's see. So this is a substrate. And now you check. So the shape of the substrate is not exactly complementary. It's not exactly complementary. But when the substrate approach the enzyme, it will induce the enzyme actually, okay, become more complementary. So in this case, how you explain? So first, okay, this is what you have to mention. So the shape of the substrate is not exactly complementary to the shapes of the active sites. You have to mention, okay, because you already observe it, right? It's not complementary, right? You can see the shape. But what the enzyme substrate can do here is, so substrates can induce the slight change in the shape of the active sites. So when this substrate approaching the substrate, the enzyme, so this even make, are you clear? This even make the what we call this the enzyme even more specific, because only the correct substrate can induce the change. So means that when you say the shape is complementary, now look at this. When I have the shape already complementary like this, can you see that? So my substrate, this shape can bind. Okay. This shape also complementary, right? It can bind. Correct or not? Or this shape also complementary. Can you see that? Because I only need this part. I only need this part. Correct or not? To bind to the active sites. Correct or not? It's complementary. But the thing here is, are they the correct substrate or not? So most of the enzyme, I mean, so with the advanced technology, when we check and look at the enzyme, actually, most of the enzyme, they are not totally complementary. It's not exactly complementary. Only the correct substrate can induce the slight change in the shape of the active sites, making it now complementary. So now the shapes of the active sites is complementary to the shape of the substrate. Are you clear? Only now they can do this. Are you clear? Then the rest, you can see that forming the temporary bonds and blah, blah, blah. Okay, form the ES complex, okay, by forming these temporary bonds, lower down the activation energy and form the products, reaction take place and blah, blah, blah. Yes, correct. But you have to include these 3 points now. Okay, when you talk about, if you look at the shape of the enzymes, okay, active sites and the shape of the substrate is not exactly complementary, then I have to mention the shape of substrate is not exactly complementary to the shape of active site, but substrate can induce the slight change in the shape of the active sites. Therefore, now the shape of active site is complementary to the shape of the substrate. Therefore, substrate can bind to the active site to form the ES complex. Temporary bonds can be formed and therefore lower down the activation energy. You can see that it's similar to what we described previously. Okay, so you can take the photo of these diagrams. So now our group of the amino acid that make up the active sites, okay, catalyze the conversion of the substrate to the products. The products can be released from the active sites and the enzyme is now free to take another substrate molecule to its active site now. Okay,